Okay, good morning. We'll go ahead and get started with our second session. My name is Danielle McLeod-Henning, and I am the program manager for the Forensic Anthropology, Forensic Pathology, MDI, and CSI R&D portfolios at NIJ. Sorry, I feel like it's really hard to breathe with these things while you're trying to talk. But I'm doing this because I don't want to be that person to give anything to anybody and, vice versa. So, but we have a really great session coming up. We're going to highlight some projects in our MDI, Anthropology and Pathology portfolio, looking into tools and techniques for applications in forensic pathology and forensic anthropology. Some of the projects that are featured are investigating novel technologies for sorting commingled remains, determining cause and manner of death in infants and children, and as well as differentiating between perimortem, antemortem, and postmortem trauma. Our first presenter is Dr. Matthew Baddeley from the University of Central Florida. So without further ado, I'll turn it over to him.
All right, good morning. So first of all, thank you a lot for the invitation and so being here. I really want to thank FTCOE and NIJ for giving the opportunity to show what we've done for the last 2 years. So this work is really a very exciting collaboration. So at UCF, myself and Kristen Livingston, who is the grad student working on this for a PhD, as well as Katie Zedlik and Nick Pasalacqua at Western Carolina, and John Bethard at USF. And so this project is really about bringing elemental analysis, especially field deployable elemental analysis for the pre-grouping of commingled remains. So the outline is just giving you a little idea where we fit in the challenge of commingled remains and why we wanted to bring a chemical basis for it. Also, what the results after 2 years. We analyzed 62 individuals, and so we'll show you everything that we've done, especially on data analysis. And also, I'd like to bring up some discussion, some points of discussion to where to go from now on. And of course, a disclaimer that this is our findings, nothing with the DOJ. So first of all, commingled remains, if you don't know, the situation is just to have intermixing of remains. And so some of the main situations is really you have mass graves. So either it's like mass disasters, natural or criminal. It could also be humanitarian forensics. So if you have human rights violation, you have war crimes, but also in bioarc. And so if you have also some examples of burial practices, religious rites, et cetera. Also, it could be that you didn't have intermixing, but then you have scavenging, or you have scattering that can be, so animal scavenging could be scattering, especially in the humanitarian forensics aspect, if you have manipulations of the mass graves. And also, you could have, due to a lack of training, and this is a big component, you could have some issues with the practices during the excavation or during the recovery of the remains. So that's a big problem, but has different aspects, and it's good to have this context to understand that when you have commingled remains, the goal, of course, is to assign and to identify each piece of osseous remain to someone. And so for this, there is no option. This is DNA. There's nothing that's going to replace DNA for identification. But our job as trying to help for reassociation is just to make sure that we can bring as many bones as possible, as many fragments as possible together. So this way, when we do the DNA, then we can maximize this amount of osseous material. And then there's just a little bit of those can be used for DNA. And we know that the rest anyway is the same individual. So that's the whole idea. So we help in having this reassociation So we can be very efficient in the identification later on. So the techniques that are used usually are physical. So there is visual pair matching, left-right skeletal remains, skeletal elements. You can have morphological traits. You can also have like joint articulation. So two bones, if they fit together and that makes sense, so you're going to put them together. Anything about alterations in taphonomy, but also more quantitative. That is osteometric reassociation. Is the statistical approach of the dimensions and the morphology of the bones that usually help more on the exclusion side. There's like we have a better idea that, yeah, those two bones really don't make sense together if you look at their dimensions. So those are physical techniques, but there's also another approach. And we're used to that in forensics anyway. There's a lot of physical aspect to a lot of trace evidence or even fingerprints. And then after, can we bring a chemical approach to it? Can we add information to help in any challenge that we see? And so this is why the chemical view of the bones is something that's well known, but just the idea is that if we have the bones, so you have the bulk, you have the matrix, that is like 70% hydroxyapatite, 30% collagen. And so if we look on the elemental side of things, you can have calcium, phosphorus, oxygen, hydrogen, and then carbon, nitrogen, just for the matrix itself. But then after, what makes it very different for each individual, and this is kind of the rationale behind everything, is that the minor elements and the trace elements that can be from different lifestyle, different medical conditions at the time of death, can kind of give a fingerprint, an elemental fingerprint, to each individual that could be a tiny differences, but maybe we can catch that. So that's the idea behind it. And we see that there's a lot of elements that we need to take into account. And the concentrations can go down to the ppm in weight. So we need something that's sensitive, and we need something that is very broad in their sensitivity. So for this, we want to use lips. And why that? There are several reasons. Anthropology is used to XRF because it's very convenient. It was the only handheld elemental instrument for a long time, and lips caught up with different reasons. But now it's very, very fast to get any elemental profile, and you have like several commercial systems. So now it's very something that is on the shelf available. And how LIPS works, it's very straightforward. You still do some sampling by laser ablation, and then during that process, you create an excitation with the plasma is self-induced by the material and the laser pulse itself. And then you have a hot gas, and then it's just going to relax, emitting light that is representative of the different elements in the plasma. So very simple. The idea is very clear. And so what we get is something like this. So we have like a spectrum. And so if you know about atomic emission spectroscopy, or even if you know about atomic absorption spectroscopy, plenty of lines for each element. And you see you can go with most of the periodic table with LIPS. That is an advantage compared to XRF. XRF is limited for the low atomic mass. LIPS doesn't. LIPS can go from hydrogen to uranium. There's no limitations. And so we can see in this, so this is an average spectrum of a bone from our own data. And we see all these elements in there. I didn't assign all the lines. That would be a mess on this screen. But we see all those elements there. So we are very sensitive to majors, minors, and traces. So we have the signal. I was telling you LIPS is used, and there's a little bit of ablation. But the thing is, if we look at the sampling, we need to have this sampling in the plasma. These are pictures on a bone. This is kind of the sampling we have from the laser pulse. With digital microscopy, we could see that it's around 150 micron wide, the sampling, and not even the thickness of a hair for the depth. So if I can challenge you to find on this picture where the crater is, It's just like, please go ahead. But just to help you, it was just right there. And so we even have sometimes issues to find them. But the idea is that it won't be an interference for at least the macro analysis of the bones, for any tool mark analysis or any trauma. But in any case, you see, this is very, very, it's just like a crater, and it's not like a striation or anything in the bone. So now we have the technique, and so we have so we can take the elemental signal, and LIPS has been used many times before us. So that was things before we submitted to NIJ. There were a few studies. So 3 studies before us, there was like 7 bones from 5 individuals, and they were showing it that they can reassociate, or like 8 bones from 4 individuals, 25 bones from 5 individuals, and then we decided to use, this was with benchtop, big instruments. So this is hard to bring the bones always there, or sometimes they have to cut them. We don't want that, so we use the handheld LIPS. And so what happened is we discussed with John Bethard at USF, and where we went is just then to the body farm there, but it was in Tampa, and then we got this now set of samples. So we had 6 individuals, close to 160 bones. Now it becomes very interesting to look at reassociation, and that gave us very good preliminary results to submit to NIJ. And so what we told NIJ, we said, okay, we're going to go further than that, and this is the equivalent of what we get to scale. So we get 62 individuals, we get equivalent to like 1,700 bones or so. We now have a lot of samples, a lot of data that we can get results on reassociation. So the samples are from Western Carolina University Body Farm, the Forest facility. That was from the John A. Williams skeletal collection. And the demographics of it, surface decomposition, only one was a burial. And then after biological sex, it's like half and half male, female, median age of 70 years old. But then on the lip side, what we have is we were targeting 35 bones per individual. Sometimes we have more, sometimes we have less than that. But in average, there was around 29 bones per individual. Depending on the size, we could get more or less landmarks. So you can see here, oops, here I go. You can see all the landmarks that we analyzed on each skeleton, and that gave us approximately just shy of 12,000 elemental profile for 62 individuals. So now we have that. Great. Let's go to the reassociation and the data analysis. And what we used was something very simple that we can control. We know what it means. And so we went with linear discriminant analysis. I don't want to spend too much time on this. The idea is really simple, is that every individual is going to have data points, and LDA helps us by maybe combining the variables that we have so that we can maximize the distance between the different individuals and making the point cloud as small as possible for each of them. This is just what LDA does. And so this is supervised. We have to train it to recognize what a cloud of points is for each class, that it's each individual. So what we used was training with 70% of the data, so 70% of those 12,000 spectra, and making sure that every class is represented the same way. And then we test on the remaining. So then we just throw a spectrum, and then we look if it associates to the right one. And what we get usually is a confusion matrix. And if we go with 62 individuals, that looks like this. So that is impossible to discuss that, impossible for us to look at anything. Even analyzing for us, that doesn't make any sense. So how can we make that more graphical and more useful for discussion? If we look and we zoom there, you see the diagonal is going to be about the accuracy to give the right spectrum to the right individual. Off diagonal, misclassification. So the idea is if we color code the accuracy for each individual, And then we show the misclassifications with arrows. Well, instead of being the right one, I was misclassified to that one. Then we start going with graph theory. And what we get now is that big table now looks like this. And so an LDA of just those samples with the spectrum not being changed or anything, no data reduction, would look like this. Still a bit of a mess. Each point is an individual, but it helps us now knowing that we have the 62 individuals and how they are misclassified. So the problem with LIPS is that you have a lot of data. Every wavelength is a variable. And so a lot of it, there's correlations, like you have multiple spectral lines for the same element. And you also have a lot of spectral regions that may have no signal in it. So how can we just get rid of it? Very simple. We go with principal component analysis, where we can just shrink the amount to what makes sense and what gives the variation of the data. We don't use an empirical or just a ad hoc rule to determine how many we choose. What we do is just we do the PCA, and then we try our LDA for each number until we reach a plateau. And then we know that if there's no more improvement, we'll stop there. And that is around 200 to 230 principal component that we can use. And then we go from just normal LDA with like no, if we have nothing to reduce the data, we can go up now. Just using 200 PCs, we can go up to 85% of accuracy. So we reduce the amount of variables, we increase the accuracy. And so what we get now we love those graphs now. We have them. And so the thing is now we see that there's way less misclassification, better accuracy. We see the average now from 60 is 85%. And even for each individual, depending, each individual has a different accuracy between 51 and 100%. All right. So we reduce the issues of correlation and noise, and we increase the accuracy. The problem with PCA, though, is that every time you would want to bring a new individual, you need to redo everything from scratch. And so the idea that we had is, can we just have kind of a filter? Can we have a mask where we say, I just want to use those lines from the spectrum. I don't want to have a combination, mathematical combination. I just want the lines of the spectrum. And so how do we do that? Well, PCA is still useful for data selection. And so what we do, if you remember, on PCA, you have all your data. You create principal components that show you the importance of each variable originally, that is each wavelength. If you just sum them up, then you can see the importance of each of them. Now, we can determine which one can I use, so this way this is going to be the important lines that I want to keep. And then this is where there was a lot of work, parametric study. How many PCs do I keep with which threshold? So there's a lot of, it's model training at this point to create that mask. And what we saw was that around 50% threshold with 200 PCs, it's just that was kind of a good trade-off to get a good accuracy. And so once we just keep what's above 50% of those of the loadings for the sum, we have just few lines of importance in the spectrum. And that represents 28 spectral lines, 1% of the data still, so we still have everything we want. We have data reduction and data selection, and there's a meaning to them. It's not just a mathematical function. So what we get is then this, and this we like that. There's so many individuals now, they are not misclassified. They don't even get misinterpreted in a way that they're not misclassified and nothing misclassified to them. We have a lot more of those, and so an accuracy that is pretty similar at 87%. Now, the point of it was to kind of see the physical meaning of this data selection. And so you see that all these lines, you see that there's like a lot of calcium line with some phosphorus and carbon. The rest is trace elements. So the trace elements and minors, we understand it. But then it's kind of a chemical fingerprint. But the calcium, the carbon, it's the matrix. It's the bone itself. So how is that helping me to differentiate between the different individuals? And LIPS is a laser ablation technique. So it's very sensitive to density. It's very sensitive to any problem that the matrix may have. So it could be, and we still work on that to interpret that, but the physical aspect of things, the state of the matrix, may be something that LIPS brings that XRF doesn't, or just a pure chemical analysis doesn't. And this is where we're still working on. So where to go from there? Well, when you have a mass grave, you don't know your ground truth. You don't know how many individuals you have. You don't know which bones belongs to what. And so we tried to go completely unsupervised with so we got self-organizing maps, and that was with Mike Sigmund at NCFS. We tried it at the beginning. It's still messy. We need more information, but that's where it's very important. What we want to do is just, can we just be the addition to the anthropologist? All right, so they know their work. We're just there to help increase the accuracy in reassociation. And so if they can help us creating that training with just the bones that they are sure it's working, and they're sure of their association, then can we use that? And just then we bring the LIPS in our approach. Then something we're very, very eager to try is to bring an error rate. So what is the error rate of bringing two bones together? So right now it's just like, oh, they do or they don't. Can we have more of a likely ratio approach that we see everywhere else? Of course, the effect of diagenesis. We all understand the problem depending on the conditions that we're going to find the chemical remains. And like I was saying, determine the chemical and physical information in LIPS. So conclusion, the chemical approach is great. It works. We managed to get 87% accuracy on 62 individuals. LIPS is a great additional tool to get in the toolkit. It brings us chemical and physical information. And we even brought a new way to see the LIPS data selection and trying to give more meaning without losing the efficiency of PCA. Now we can give it a real meaning with the real data. So I want to thank NIJ for funding us. Of course, there was the undergrads at Western Carolina. So there was Grace, Autumn, Molly, and Nora that helped a lot Kristen to do all this data. There was 15 individuals per week for 4 weeks. That was a lot of work. And of course, the National Center for Forensic Science, where we do the research and they support us. The group, of course, that here we have, like right now, there's Kristen and Caitlin who are here this week, and plus several undergrads who help in the projects. And so I want to thank you for your attention, but please, more stuff that has been from our group. So tomorrow, for sure, in the posters, I already invite you also with the toxicology posters, if it's late. But Caitlin just got a grad research fellowship from NIJ to develop hair standards for chemical analysis. And she's entered in the competition for Emerging Forensic Scientist Award as a poster presentation. Thursday is a big day as well. We have with Xenia, Nick, we have a double session on mass graves and humanitarian forensics. And Kristen is going to give a complimentary presentation of what I showed. And she even got recognized by FSF, and she got a student affiliate scholarship to come here. I want to thank you and answer questions.
Hi. Have you also tried it with some animal bones? Because a lot of times you also have some animal remains mixed in.
So this is the big question. So for us, so the context in which we work, we are not looking at human versus animal. So for us, it's just reassociation. So that would make a different group. Then after, but this is in we have another project when we're going to start looking at that. But it's like, can we see a big difference or not? And the difference is not that big between humans and animals. We haven't done for an NIJ, but we have another project working on that. Yep.
Hi. I was curious on your testing and training. Did you take bones? Did you have bones from every possible source, or did you leave some? Did you have some tests that weren't in the original training data when you built up your PCA LDA?
Every class was represented in the training and in the test. So we made sure that the 70% were already 70% from each class, and 30% was the remaining for each class. I don't know if I answered your question. Okay.
Hi. You may not have figured this out yet based on your last slide, but when you had inaccuracy, have you figured out, are you getting false negatives, meaning that the individual range of your spectra is so small that you couldn't classify, or is it the other way around? You were getting false positives.
So if you see, I don't know if I can go back to the slides, just to show quick.
On your reduced PCA,
Anyway, I mean,
on your
we can
reduced
discuss,
PCA.
but the thing is, you see that there's like 2 hours, right? Sometimes.
Right.
Just very quick. Here we go.
Yeah.
So you see when you have like 2 hours, there's going to be like individuals, like it's going to depend which
That's
way
what I
they
thought.
go. So you're going to have both. You got false negative and false positive. But then we've tried different things, and Kristen may talk about it on Thursday, where when you have bones and you take a lot of them, a lot of spectrum on them, you're going to have more chance for having an error on this. But we've done other data analysis where we show that we can manage around that. So the error is really, at this point, we're really still pinpointing where the error comes from. And so we're working on that.
Okay, sorry, I'm up next. Am I next? Oh, okay, sorry. Great talk. I confess I missed the first few minutes, so this may have been covered. I noticed when people have diabetes, that their skeletons go brown, and so pathologies such as that could affect your readouts quite significantly, potentially. And the second question is, with the, and you may have covered this, with your carbon-nitrogen ratios, a lot of people spend time have you used that as a way to filter for high-quality preservation and bones that could be used for proteomics or DNA analysis?
So we haven't looked at having additional data analysis for the state of the bone or preservation of the bone. I know that the carbon-nitrogen ratio is sometimes used, especially if to see like the part of collagen that wouldn't be there. Here, all being from this, it's kind of modern, and they're all kind of the same, so they will own surface decomposition as well. We considered that was not so much of an issue, but that's something we can always have a look at. We have all the data now, so we can have a look at that.
All right, we'll take our last one from Natalie.
Okay. Hi there. Yeah, that was a fascinating talk. I'm just kind of curious about what the main drivers of differences in trace element composition would be for bones. So I guess I'm imagining a mass casualty situation that's like a plane crash where the people on the plane are all unrelated individuals who all grew up in different places and drank different water and whatever, versus what if the plane crashes onto a small village and all the people in the village eat the same food, drink the same water, et cetera? Just sort of, if you could comment on that.
Yeah, but the more different the people could have a different profile, the easier it's going to be for that approach. We don't solve everything. So the idea is really, that would be nice way, like nice future studies, of course. And then even elemental, we can always go with isotopic. I mean, that's going to be the best way, right? So there's a lot of work to do. I mean, I'm very happy that NIJ-funded us to start this. Now it's just we need to build up and bring the community to get more of those questions answered. Yeah, that's for sure. Thank you.
And we'll welcome our next presenter, Dr. Jennifer Pakal from Michigan State University.
Good morning. So I'm going to, I guess, represent the pathology side of this section, since there seems to be quite a bit of the anthropology and the different presentations that you're going to hear later, and then the one previously. I'm going to talk about the practicality of the postmortem microbiome and death investigation, specifically talking about infants. And so this is kind of a unique area because to date, in all honesty, we haven't had a lot of infant work. So this has been a unique opportunity and kind of an interesting time, especially because I have a 15-month-old, so it's been a wild time having this project at the same time as raising an infant. So just because there is a wide breadth of knowledge here, we all may not be familiar with the terminology that I'm going to use. And so what I'm going to talk about here is kind of the different phrases that you're going to hear commonly when it comes to at least the microbiome. And so there's a lot of different terminology, and I am 100% guilty of this, where people are trying to name everything a unique kind of catchy name for the microbiome for whatever system they're studying. The one that we came up with probably about 5-ish years ago is called the necrobiome. And the necrobiome was actually talking about all of the different organisms that are associated during death. So this includes things like insects, vertebrates, microbes. But that term has kind of been co-opted to just mean the microbial communities that are associated with death. And so that's not necessarily what I refer to whenever I say the microbiome or the necrobiome. And I talk specifically about the microbiome of the necrobiome. So lots of biomes. And so one of the things that I wanted to talk about is that the microbiota, that's going to be the different actual organisms that are present in the system. So that may be a term that you hear frequently when people talk about the postmortem microbiota. And the microbiome is going to encompass all of the different genes that are present. So it can be a wide range of things. And so you're going to hear the word microbiome quite a lot. And that's something that's also going to deal with the structure. It's going to deal with the nucleic acids. It's going to deal with the proteins. So it's going to cover basically everything that's within that system. And obviously, you can have microbiomes at different levels. So you can look at very specific systems. One's talking about, let's say, a specific area on the body, or you can think about it as a whole ecosystem. So a very wide scale about what your information you're garnering from that different topics that you're actually analyzing. And so today, whenever you hear me speak, I may say microbiota, I may say microbiome, but in all actuality, it's just talking about the microbes and their function that's happening in postmortem intervals for infants. So one thing that you may have a question about or may not necessarily think about often is, what is a microbial biomarker? And so biomarkers are specific regions of typically DNA. And what we look at a lot of the times is a 16S region. And so this graphic that you see on the screen right here is a representation of the 16S marker, and that is a very commonly used region of DNA of microbial markers because it is conserved across a lot of different species, basically all of them, for both bacteria and archaea. And so when you look at these different regions, there are different ones that are going to be conserved. They're going to be highly variable regions. And so you can look at a lot of different areas. And so one of the things that we look at is just specifically the V3, V4 regions. Those are the ones that are typically the most commonly used when analyze the 16S data. And so some people like to look at the whole region, and that gives you a very, very specific answer for what species are present on that body. The V4, V3 regions, they kind of have a wider breadth of information that you can garner, and it's not going to necessarily be as specific, but you can still get a tremendous amount of information. So this is just kind of an example, and there are lots of different microbial biomarkers, but this is, again, one of the most common ones that you see when you hear people talking about the postmortem microbiome. Now, thinking about the postmortem microbiome, this has been going on for 10, 15-ish years now. I guess after COVID, like, my time scale is completely off, so probably closer to 15, 20 years. And so one of the things that we've identified as a group collectively, because there are lots of different researchers that are focused on this particular topic, is that you can use the microbial markers as a way to identify how long an individual has been dead. You can identify specific regions on that body, and then you can also kind of identify ante-mortem lifestyles. And so these are all really important questions when you think about death investigation. And a lot of work has, or I should say, a lot of tremendous work has gone into this. And you're looking at people who have worked at body farms, you're looking at people who've worked at medical examiners' offices, and so you're really getting this wide scale of information about the postmortem microbiome and how that information can be used in depth investigation. So for this particular study, we have this kind of preset criteria of specific areas on the body that we're interested in. Excuse me. And so the criteria for this particular case is that we looked at individuals who were around 2 years, so toddlers and below. Really, it was difficult to get individuals over the age of 6 months. That was our least, the least number of cases that we had. And then for this particular one, we had males, we had females, and then we had black and white individuals. And so these were all bodies that were being autopsied at the Wayne County Medical Examiner in Detroit. And what we had was a wide scale of different manners of death. Excuse me. And one of the things that we were interested in was obviously how these individuals died. And so what we were looking at were all of the different causes of death, the manners of death. We had obviously 4 different types. We had accidents, homicides, we had natural deaths, and we had indeterminate deaths. And so we had a lot of different variability whenever it came to the ways that these individuals died. And I have a list here too about the specific causes of death, if that's something that you are interested in. There's a wide range, again, of the different types of ways that people died. And so we have this routine where the pathology assistants or the autopsy techs, they'll sample the bodies when they're doing their autopsies, and we want them to focus on very specific areas of the body. These are areas that we were looking at in adults, and we saw very distinct differences in the postmortem microbiome after death, and these differences were very specific to the body regions. So we were curious, did this still apply to infant cases? And then what we did is that we took those swabs, processed them in the lab, and then we went through a whole different spectrum of different analysis and techniques. And so we extracted the DNA, quantified it, amplified it, and then actually used 16S Illumina sequencing, and we also looked at whole genome sequencing. And so all of that different data were analyzed, and we used a lot of different open source bioinformatic pipelines. So these are things that anyone can use, and it's not going to be something that's paid for, but it does have a little bit of a learning curve. So these were the different kind of approaches that we had to identify what the microbial structure and function was. So for what I'm presenting today, it's a total of 44 cases, and that may not seem like a lot, but if you think about the number of infant cases that an individual medical examiner's office may receive in a year, we felt that this was a very strong number because, again, we had a tremendous amount of diversity that was also associated with it. And so we had 44 cases. You can look at the composition. About the male to female ratio was about 50-50. We had more black cases than we did white cases. In terms of the manners of death, the most common manner of death that we had was going to be an accidental death. A lot of times the accidental deaths were co-sleeping events, and so these are times where asphyxia occurred and the individual, it was in an unsafe sleeping environment, and that led to its death. And you also had homicides, we had natural deaths, indeterminate deaths, and then we had a few cases that we were still pending on our information, and so hopefully we can fill those gaps soon whenever we get the actual causes of death. Now, I know these are not fun to look at, but for those who are interested in terms of age structure, this is kind of how it breaks down in terms of the manner of death as well. So we have a lot of cases under the age of, again, that 6-month range. So that's primarily what I'm going to focus on whenever I talk about a couple of different results here in just a minute. And again, we had males and females. It was relatively similar in terms of their structure for what cases we were sampling. And again, we had more black individuals than we did white individuals. And this is a random sampling of what was coming into the Wayne County Medical Examiners, and so we didn't have a bias as to selecting one race versus another race. And so this is just kind of a representative sampling of the population of Wayne County. So in terms of sequencing outputs, I think everybody who does omics likes to put how big your data set is, just to show like it does take a little while to do some sample analysis. And I'm just talking about whole genome sequencing for this particular data. We also have 16S, which I'm going to talk about later this week, and I'll plug that at the end of this presentation. But for the 16S, out of our 44 samples, we have about 1.8 billion reads. This is a lot of data. It's a lot of noise. It's a lot of information that you have to parse through. And so for each case, we had around 40-ish million reads per individual. And just to kind of give you context about that, when you're looking at 16S data, so looking at just one specific biomarker and not everything within that particular genome, 16S typically produces about 10 to 12 million reads per sample. So you're getting an exorbitantly amount or a greater amount of information from the whole genome sequencing. And you can see there's a lot of variation for individual cases. Now, just to kind of give you an example of some of the different work that we were looking at, for the functional analysis, this is just a heat map of those individual functional identifications that were hotspots. And so we have about 120 different gene families that we're going to be processing and going through. And so this is just a snapshot of 0 to 6 months. And we were looking at the individual biomarkers that were present in all of the cases. So this is a very small number, so the prevalence had to be 100% to be included within this heat map. And what you can see is that there's some groupings, and really what we're gonna be focusing on is parsing that down and really reducing the data, the noise that you're seeing in these particular data sets in order to get a clear signal to hopefully identify how the individuals or the different manners of death are influencing these functional heat maps. And so just to give you a very quick example about a certain kind of like the translated protein search, these are just a couple of different ones that were popping up in all of the different cases, so I thought these were interesting. And when you look at the individual biomarkers across all of the cases, and in this particular example, this is manner of death, or excuse me, this is manner of death. And so we have the natural cases, then we have homicides, or excuse me, we have the accidental cases, homicides, natural cases, indeterminate, and then no data. And so you can see that there could potentially be trends to identify how individuals were dying based on these biological markers. But we need to, again, confirm and validate these different biomarker signals in order to determine if they're true signals or if they're just noise. So one of the things that's important to consider with the practicality of the biomarkers of the microbiome is that you have a tremendous amount of variation that exists within individual populations. This is something that was discussed in the previous presentation. And so thinking about those microbial signatures, we really want to identify those that are going to be consistently present across all of the individuals that we identify. And then also we're going to be able to identify what those individual profiles are and if they're going to be consistent across all the different cases. So really, one of the things that I wanted to just finish this presentation on is that kind of the best practice recommendations moving forward is that this is a working group, or it's not really a working group that's official, but this particular group is really focused on how to standardize these processes, because the way that I run things in my lab is going to be different from how other people's run things in their lab. And so we really want to be consistent in kind of the processes, even though each individual step may not necessarily be the same. So I think it's going to be really beneficial for us to standardize our protocols, kind of integrate the ways in which we are actually identifying these microbial biomarkers. It may not necessarily mean having the same pipelines, but it's something that you're going to be able to routinely use within your forensic analysis. And then finally, we want to be able to collaborate. And so I think collaboration within forensic sciences is kind of like this beautiful thing, because you get this interdisciplinary approach. You get lots of different people who are incredibly smart, and they have very different perspectives and can inform what you're doing. So I think for the projects that I like to focus on, I learn a lot, and I realize that a lot of people are incredibly intelligent, and it's something that's incredible to see kind of how one thing can influence lots of different aspects of the individuals who are working in different disciplines. And so with that, I think as we move forward, we're obviously going to continue to analyze a bunch of these data. We're going to hopefully identify some key biomarkers that are going to be consistent across all of our cases. And I really think that we just need to continue to collaborate and reach out to other people who are in different disciplines who may have different perspectives about how you can use these types of data in order to identify the questions that are going to be important and relevant to death investigation. And so with that, I just want to thank NIJ. I want to thank everybody for the invitation to participate here today, and obviously all of the collaborators who are part of this. And then I will also talk about this a little bit more on Friday afternoon for all of y'all who continue to stay through the week. So with that, I will take questions, and we're kind of on time. Sorry. Try to go fast.
Hey there, Dr. Peckle.
Oh, hi.
I might have missed this. What was the sample type?
Oh, they were swabs.
Of like fecal or skin or?
Oh, I'm sorry. These were all different areas on the body. So it could be ears, eyes, nose, mouth, rectum, intertrabecular space, and the interhemispheric region. So it was basically external, and then we had a few internal for the brain.
So I was just kind of wondering like what kind of hypotheses you're thinking about, because I was just thinking about for something like RSV or something where an infant was really sick, you probably would see that marker in the gut, as in, because usually you see sort of a massive drop in diversity with severe illness. Like, do you have others of those types of hypotheses about the body sites and the biomarkers?
Yeah, so I think with I think the nose is going to be an incredibly important one, because think about that, that's kind of a filter to your outside environment. So if we were to be more specific towards viruses, I would need to bring a viral person way outside my area. But I think that your nose is going to be something that's incredibly important. It's kind of interesting to see as the postmortem microbiome changes over at least decomposition time in adults, you do see this distinct change in the succession patterns of that individual body area. I also think the brain is going to be kind of an interesting one, and it's one that we're really thinking it may not necessarily be as sterile as what everybody thinks, and that's something that I think is going to be incredibly important as we move forward. And again, kind of like your external surfaces that are kind of just being exposed to everything. It's very interesting to see how those are different, even with individuals who die in similar manners. So those are the areas that I think that we're going to continue to focus on.
We have a question from online. How does the heat map of 30 million reads relate or translate to the number of microbial species? And in parentheses, sorry, my microbiology knowledge is still stuck in genus species.
No, that's a completely excellent question. So the heat map that I demonstrated was actually functional, and so it could correspond to many different species that are performing that function, or it may correspond to one species that's performing that function. I don't have the exact answer for that. However, you can use microbial heat maps also to map your individual species levels. I just didn't show that in this particular presentation because I'm sure y'all don't want to be here for 6 hours hearing about the microbiome. And then there's one question over here.
Thank you so much for the presentation. Just out of curiosity, now the metadata that you collect from these individuals, how can you, like the variability of these metadata and correlating it with the microbial signature, like the cause of death, for example, if this is one of the variables or metadata that you've collected, if this is not accurate, how can that be correlated to the microbial biomarkers?
That's a really good question. So we trust our pathologists and their opinions. Obviously, there are pathologists that can have different opinions as well if they were to interpret specific data, or let's say the slides looked a little bit different to them. One of the things that you can do, and again, it's just we could talk about this all day, is that you can basically map where your microbial signatures are in relationship to each other. So for my no data examples, I can see where they're pulling out amongst the other individual cases, and maybe perhaps that's going to line up to an accidental death, maybe it'll line up to a homicide, maybe it'll line up to a natural. There's a lot of different variation that occurs within each of those individual types of manners of death as well, so sometimes it's a little ambiguous. So I understand what you're saying in terms of if you have a different pathologist, you may have a different opinion, and that will influence what your outcomes are going to be. But when you look at the actual raw data, we could also identify maybe there's a cluster over here, maybe there's a cluster over here, so we can investigate a little bit further as to what may be driving that specific clustering pattern.
All right, I think we have 2 virtual questions, and then we will wrap up.
Question from online. Since the microbiome varies with postmortem interval, what were the PMIs of the cases studied?
Oh, I apologize. These are all less than 24 hours postmortem.
Another question. Are one of the biomarkers related to the different nucleases?
Oh, that's a good question. I don't have an answer for that. One, biochemistry, surprisingly, not my strong suit. I'm still an entomologist. Great. Thank you so much.
Thank you so much, Jennifer. And our next presenter is Dr. Brittany Coates from University of Utah.
Thank you. Hi. Start. All right, thank you very much. As she said, my name is Brittany Coates from University of Utah. I'm going to talk today about age and sex-based skull thickness distribution in the infant skull. So we're maintaining the infant theme, which I'm actually delighted to see. What I'm talking about skull thickness for is really in the context of how it influences skull fracture patterns in infants. So first, my acknowledgement and disclosure. This work was funded by the National Institute of Justice, and the opinions, findings, and recommendations are those of mine and not the Department of Justice. So I guess it wasn't previously mentioned, one of the motivations for the work that I do in my lab is child abuse. Child abuse has and continues to be a prevalent problem in society. Infants in particular, less than 12 months of age, are highly susceptible to child abuse. They have the highest incidence of death from child abuse and the highest likelihood of reoccurrence if it doesn't lead to death, which then increases the risk of death. So it continues to be a prevalent problem. As you saw in the previous talk, the younger infants are the most susceptible to child abuse, and so that's a lot of what we study in our lab. Most of the times, there's no witness to the event that happened to the child when child abuse is involved. And so you rely on the injuries to tell you the story of what happened to the child. The typical story that's provided is the child fell. There's a lot of accidental falls in children. And so trying to distinguish between accidental and inflicted trauma is actually an important piece to understanding what happened to this child and being able to identify truthful histories from false histories. One of the injuries we've been looking at is skull fractures. Now, skull fractures are common in both accidental and inflicted trauma, so why are we interested in them? Well, there's a lot of other injuries that can happen in abusive head trauma. However, skull fractures, we know there was an impact, so we're already one step ahead of the game on what caused it. There was some kind of impact to the skull, and actually the fractures are physics-based. So if we can understand the mechanics of the infant skull and what drives fractures and crack growth and directionality, we can understand a lot about what happened to the child and tease back the history and the story that likely led to it. So to get us to this point, we previously developed a computational tool set to predict skull fracture patterns. This was a previously funded project. And so we used fundamental fracture mechanics, which surprisingly is not used often in the bio realm. It's used often to predict crack growth in airplanes. So there's actually quite a strong literature behind fundamental fracture mechanics. It's been around forever. So we took that knowledge and we implemented it into an infant head model to predict crack growth from accidental fall scenarios, with the premise of if we know what can occur or probabilities of injuries that can occur in accidental scenarios, that gives us one step closer to either verifying a history or discrediting a history. Our previous model was based on a 1.5-month-old infant. This is an example of a simulation. Let's see if this works. Nope, it did not. Is there a way to get the movie to go? There we go, perfect. There we go. So we're actually predicting the crack growth, and it's highly dependent on the impact location, the angle of impact, the height of the fall. So all of these factors go in, and our model can predict both the initiation and the propagation of the crack. We validated it against several cadaver case studies where they had documented skull fracture patterns in infants, and it did quite well. However, we realized that this is just a single model of a single infant head. It had non-uniform skull thickness, which was great, but it's just from a single infant. So how likely is it that another 1.5-month-old infant is going to have the exact same skull fracture pattern given the exact same impact from the same height? And that's one of the things we wanted to ask. How generalizable is this model? And we know, as I'm a mechanical engineer by trade, so I know that skull thickness is going to determine at least how far it grows, the length of the fracture, and probably some other things as well. And so one of the things we wanted to do is move our model to more generalizability. Anytime you have a computational model, you have to build in the variability. You have to build in the uncertainty. You can't just simulate a single case. You have to understand how it varies in a lot of different scenarios. And we wanted to focus on how does skull thickness distribution affect predictions of fracture in our model. We wanted to also understand how does skull thickness affect it with age, and are there different skull thickness distributions and thicknesses between males and females? So how does age and sex influence our skull fracture patterns? Are there ages that we can group together so that we can have a single model represent a certain age or a certain sex, or combine both sexes so that we can have some kind of anatomical standards for predicting skull fracture pattern? Or do we need to have individual models for every infant that we come across? Can we also, is there symmetry involved in the skull when we're modeling the skull anatomically? And the reason why we ask this question is because we see it a lot actually in our realm, where people will develop a model based on one half of the skull and just mirror it to look at the other half. And is that a valid assumption to make? So we wanted to ask that question as well. So skull thickness, I should state, we are not the first group to look at skull thickness distribution in infants. There are papers out there. Some are really small data sets, so really maybe 1 to 2 cases per age range, and then for 12 months and younger, maybe about 30 cases, so not a good distribution. But there's also papers that have really large distributions. They're really large populations that they studied, but the challenge is they have also wide ranges, so they group 0 to 1-year-old infants. And in a study that we previously did, where we looked at what are skull fracture patterns in infants and how do they vary from age, we actually broke up our infants into 0 to 4, 4 to 8, and 8 to 12-month-old ranges. And I think the takeaway on this slide is really the bottom plots, where the skull fracture pattern and the characteristics are very different for the youngest infants than from the 4 to 8, than even from the 8 to 12. They have different thicknesses, they have different patterns of injury, different characteristics, their bones are responding differently. And so really we can't treat them as just a single entity 0 to 1 years of age. We have to understand it throughout. And so the objectives of the study that I'm presenting today here is really to develop, first of all, an imaging pipeline. Can we take CT images of kids' skulls, of infant skulls, and automatically extract the skull thickness distribution in a high-resolution manner? And so that was one of our goals. Also, if it turns out that skull thickness plays a huge role and we can't find age groups to cluster, then we need a pipeline to actually look at skull thickness distribution in everyone, right? And to do individual simulations. So this would help us develop that kind of backup plan if we need it. We also wanted to identify data-driven age groups. So you saw in my previous study that we just broke it up 0 to 4, 4 to 8, 8 to 12. This happens all the time. Your kind of a priori definition of age. But I was wondering, are those classifications really appropriate? Where should we be drawing the line for the different age divisions, looking at skull thickness in particular? And so we wanted to do this with our data set to see what are our categories that we should really be looking at. And then, of course, we want to look at how it varies with age and sex, skull thickness, that is, and then test that symmetry assumption. So this is just an overview of our imaging pipeline. I'll go into the different pieces individually, but really we just start with radiological data, segmenting it, cleaning it, aligning it from all 3 dimensions. That's actually critical, is to get all 3 views into your data set, and then develop these high-resolution skull thickness kind of maps that you can actually look at how it varies across the infant skull. So our database that we got the CT images from was from Primary Children's Hospital at the University of Utah. We had 288 infants less than 13 months of age. We did go back and check their ages, and we did find some errors. And so we actually have a single 14-month-old that was classified as a 13-month-old. So you'll see they're kind of lingering in the data set. We took images from axial, coronal, and sagittal views, and we had to do some pre-processing where we clean it up, remove noise, remove artifacts, and all sorts of things. We also cropped our data collection at the brow line and back. Most of the impacts we're interested in are to the upper part of the skull, and so that's really where we were focusing our thickness distribution. We segmented the images, and then we used a custom code to actually radially sweep around the skull to get the thickness. And we wanted to radially sweep so that we could get that radial thickness as we're doing it. In our bottom image, you'll see that we aligned all three views, and you'll actually see these strips. And the reason why we have strips is because you can imagine that if you're taking any kind of sectioning as the skull curves, your thickness becomes inaccurate, right? Because the sections are no longer tangential to the plane that you're cutting is no longer tangential. So we took a really conservative approach where we really trusted just the data in the strip, and we relied on the other views to provide the missing information. And so that's why 3D views is actually important. So this is what we ended up with. Each infant had about 15,000 data points of thickness. So a really high resolution anatomical map. You can imagine how hard it is to run statistics on that. You can run statistics obviously with age, but to do grouping and interaction effects and all of that. So we did reduce the data set down to 132 locations. So we averaged our thickness values across these 132 locations and used just the average values at each of those locations for each of the infants. This resulted in 50 parietal bone locations, 22 frontal bone locations, and then 10 occipital bone locations. And we looked at age, sex, and bone. And first to define, now I did run the data continuously, and of course you'd imagine skull thickness increases with age. I think that's kind of a known value. So we wanted to start with how do we define age groups so we can start looking at interaction effects and do some ANOVAs and so forth. So we used Jenks natural breaks to identify where are the natural breaks in skull thickness alone, regardless of age. Let's kind of set age aside. Where are the natural thickness changes in our data set? And we found that 5 groups was the best way to classify the data. Any more than that, you don't get any increase in accuracy. And so you can see the different colors of the different groups that we divided, and they're lateral because they're based just on skull thickness, but you can see they span a wide number of ages, which is normal in biological studies, right? Skull thickness varies from person to person. So then we needed to figure out, okay, how do we now bring age back into this? And so we wanted to define the intervals at which our age groups could best capture each of these groups that we defined through the Jenks optimization. So we used a receiver operating characteristic analysis where we really swept through all potential upper and lower images or upper and lower limits to identify the range that best captures each group. And you can imagine that resulted in overlapping age ranges. We didn't want to restrict it right away because we'd bias our data. If we said, okay, sweep from 0 to what upper limit and then start at that upper limit and continue, what happens is you bias your data because if you start at the top and work your way down, you're going to get different answers. And so we just left it open, saying where is the best upper and over limit, regardless of whether they overlap. But then we had to eventually correct the overlap because we have to make a line, right? We have to bin them. And so what we did is we created our own optimization routine, which then when they overlapped, we swept within there. And with that, you're creating 2 groups. And so we optimized to minimize the variance within the group and maximize the variance between groups. Because at the end of the day, your group should be statistically different from one another, essentially. And so by doing that, we ended up with 5 age groups, okay? And the numbers, the breaks are not going to be pretty, right? 0 to 1.8 months, 1.9 to 5.4 months, 5.5 to 8.3 months, 8.4 to 10.3, 10.4 to 13. Not pretty numbers, though you could probably round these up if you wanted, and it would probably you wouldn't get much error. So 0 to 2, 2 to 5.5, 5.5 to 8, 8 to 10, and 10 to 13. So the first takeaway from this is when we binned, in our clinical study, when we binned infants with skull fractures 0 to 4 months of age, that's 2 groups here. So perhaps those weren't the best bins to make in that previous study. So it's kind of food for thought on why we select the age groups that we do. So using this, I'm going to just kind of give an overview of some of the things we found, and then at the end I'll talk about how this fits into a broader context. So as you might expect, frontal bone was significantly thicker at any age, regardless of age. Occipital bone was the next thickest, but not as thick as you might think from the parietal bones, and maybe because we cropped at the brow line. And then, of course, the parietal bones were the least thick. And sometimes the occipital bone was significantly thicker than the parietal bones. It really just depended on age. So what does this highlight? It just highlights different rates of growth for the bones themselves throughout the span of early development. Some of the really interesting things were the sex-related effects. Males and females were significantly different at every age group. And oftentimes the males, and actually most of the times the males had significantly thicker bones than the females. However, this was with the exception of the earliest age group, the 0 to 1.8 months. Females had significantly thicker bones in that really young age group. And so what that meant is that the rate of growth of skull thickness was greater or faster in males compared to females. And so it really lends itself to say that there are differences between male and female infant skulls, and that we need to make sure that we capture this in our models or account for it somehow. The symmetry analysis was also interesting. To perform the symmetry analysis, we essentially did paired t-tests between the left and right parietal bone. We had one location on this side paired to the exact same location on the other side. And within our age groups, we did pair analyses to see if there were significant differences between the left and the right. And what we found, what you see is the metric is how many of those locations were not significantly different. So that's what the 60% means. So in the youngest age, 60% of the locations were not significantly different within an age group, but 40% were. And that number of what was significantly different within an age group decreases as you get older. So that the symmetry of an infant's skull was actually not a great assumption at the younger ages, but probably gets better as you go through age. So again, some of those age-related effects. So just the conclusions of this small study was that we identified appropriate age divisions. So actually, I think in my abstract for this talk, I defined age divisions. This is something we actually did at the end because I just wasn't comfortable, and so we actually defined these age divisions. And I really do think that's a strength of this study, is trying to identify what, in this time period of rapid development in an infant, what are appropriate age divisions that we really need to be looking at. We did find sex and age-based differences. And so this suggests that we need to consider this when trying to develop computational modeling tools and look at skull fracture in infants, and that we will have to have sex and age-based anatomical standards if we want to develop some kind of standard modeling approach. Now, the developed pipeline we have allows us to do infant-specific modeling. And so one of the things I want to talk about is I have a student that is currently, or he just finished working on an initial similarity metric to try to identify within the age group that we've identified these breaks, is there an infant that has a representative skull thickness distribution that can be used for everyone else in that group? What is the most representative infant skull that we can use as our anatomical standard for modeling moving forward? Using a specific infant is better than averaging over a case, because when you average, you tend to lose some nuances. And so we want the most representative infant, and we've identified that. And he's further working on the similarity metric to try to identify if a new case comes in and a new infant, how similar is that infant to this standard, and can we use it to simulate the infant? Or are they an outlier? Do they not quite fit in this group, and so they need to be modeled individually? So that's one of the things that we're currently working on. And right now we finished the skull thickness study, and that data, all the 132 location data, is being made publicly available online for all of our cases, as well as with these anatomical standards or the the case that was the most similar to everyone else within their age and sex group. So those are being made online right now, and the paper's been submitted. So putting this into kind of a broader context, or being able to translate this, now we have these skull thickness distributions that are at all these different locations. And so we have this distribution of skull thickness across these 288 cases. Now the ultimate question is how does it affect our predictions of skull fracture pattern? If we simulated all of these cases, is the skull fracture pattern pretty much the same, right? Maybe some small nuances, but pretty much the same. And so we did a preliminary study on this using other data, not our data. And we found that actually oftentimes it's the same and sometimes it's different. So this was just a random sampling from a previous group's skull thickness data. Again, pretty coarse selection of changes. But you can see that there are differences in skull fracture patterns. There's some similarities between the patterns, and there's also some differences. And so now the next step, what we're doing right now, is we're taking these distributions and we're sampling from all the different distributions to create a variety of models. And we will simulate how the exact same impact results in a variety of skull fracture patterns. So what is the variability within just a single impact? And that's something we're working on right now. And then the other thing is how are we going to use this model going forward, right? Not many people will want to use a model or have the expertise to use a computational model like this. So how do we make it more accessible or more usable? The great thing about our computational data set is we know exactly what the input was that led to the output. So we have a direct link to what was the height, what was the impact angle, and what was the skull fracture pattern. Whereas in the real world, you just have the output. You don't have the input. You have to estimate what the input is. And so it's a perfect opportunity to use machine learning on our simulation data. So we can simulate all these different scenarios, all these different falls, impacts, train a model to recognize the fracture patterns that result from these inputs so that it can estimate what's the probability that this skull fracture pattern had a certain height or had a certain impact location, and how do we back that out? So they just submitted this is a parallel effort running in parallel to the thickness study, and they just submitted their first paper on this. It's still in review, so can't say that it's been published yet, but this is just an example of one of their outputs. They've been looking at a variety of models, like what's the best way to do this prediction, this link. What you're seeing here is a Gaussian process regression model. And what I really love about this model is it builds in uncertainty in the prediction, right? Every model is going to give you an answer, but we need to know what the confidence is that we have in this answer. So you can see that this is a prediction of impact location. The blue dot is the prediction. The yellow goldish dot is actually where the actual impact was. And so it does a pretty good job. And then the blue cloud is how confident is it in its prediction. So a really big blue cloud would be not confident. A smaller cloud would be more confident. So this just gives you a sense of where we're heading with this. I just want to quickly acknowledge everyone who contributed to this work. Yusuf Alsania, he's the grad student who's done all the skull thickness work and is working on the similarity matrix. Ashley Spear, she is a fracture mechanics expert. She looks at crack propagation in airplane wings and in metals, and I pulled her into this project, and so she's been phenomenal. She's working she helped to develop both the model as well as she's working on the machine learning model, because that's her other area of expertise. Tigrid Ruiz Maldonado is our clinical collaborator, and Jacob Hurst is a graduate student working on the machine learning. And of course, I want to thank the National Institute of Justice for funding this work and funding more work on the mechanics of injury in infants. And I'm happy to take questions.
Thank you, great work. One of the truisms of forensic anthropology is that there's very little difference in non-adult skeletons with biological sex. So you've seen a difference in your work, and you've been talking about prediction of fragmentation patterns. But using your same data set, have you actually tried to go back and predict sex?
Predict sex from the fracture pattern. So, I mean, this is the first time.
Sorry.
So go ahead.
I'm not. I wasn't clear. Predict sex from the thickness of the skulls.
Oh, interesting. Yeah, we haven't done that. We haven't gone back to see, can we look at the skull and then predict the sex? That is an interesting question. There's so much variability. I think that's one of the challenges. And so there's always going to be outliers, but it would be an interesting question to try to address to see if we could go backwards.
If you could see a difference, a lot of people would be paying a lot of attention.
Okay. I will look. I'm going to go back to our lab. And we're going to look at that. All right. We have here, and then here, and then we have 4 online. Okay. And then we got to move to our last presentation.
How is prematurity handled with the age brackets in terms of measurements and looking at what the gestational age was versus living age and what have you?
Yeah, that's an excellent question, and one that we've gotten as well. If they were less than 2 weeks or more than 2 weeks premature, we did not include them in our study. The ages that we reported were from birth, though, and not from development. Yeah.
I know your emphasis was on skull thickness, but in your models, when you're looking at propagation, are you including sutures at all? I couldn't really tell because that's huge for propagation.
Yeah, so our model is a full head model, so it does have sutures. The skull fracture mechanics, so we did a lot of preliminary testing on what drives fracture in infant bone. And so a lot of the fracture mechanics, it's going to end at the suture. It's not going to cross over. We don't know how energy transfers across suture. So our predictions of fracture patterns are focused on the bone, but the model itself incorporates sutures because of the deformability and all of that. So that's how that's included, but not necessarily how energy transfers over suture. Does that answer your question? Okay. All right, 4 online, and then we'll go to our last presentation.
One question. In cases where there is possible neglect, there may also be malnutrition affecting growth. How might this affect which skull thickness distribution to use?
Yeah, I mean, that's an interesting question. We haven't, all the infants that we've looked at are normal infants, so they don't have any diseases or abnormalities or malnutrition. I think that is an interesting question to see how that could affect it. Obviously, there's the geometry changes, which I think our models could capture, right, if we knew what the thickness was of the skull. However, if the functionality or the fracture propagation could potentially change in like weaker skulls, if those mechanics change. We don't know that answer yet. It's a good question.
Some of the other questions were already covered in the earlier questions. Another one, though, how did you average skull thickness across sections, and what was the standard deviation of thickness for the different sections?
So across the different maybe locations, yeah, so we basically defined a radius around our center location and then took that radius as our average. I don't have, off the top of my head, the standard deviations of the locations, and part of that, the reason why I don't have the standard deviations of the locations is because we're actually taking the entire distribution curve to move on to the next step. But I just don't know how to answer that because I don't know quantitatively how much. And I'm an engineer. Everything has to be quantitative. Okay. Well, thank you again, Brittany. Thank you. And our last presenters, Dr. Jessica Skinner and Dr. Natalie Langley from Arizona, Mayo Clinic in Arizona. Sorry.
Yeah. All right, great. All right, thanks for staying. We've been here a little while. Would you guys like to stand up and kind of stretch for a second? Yeah. Feel good? If you want to, please do. You're not offending me. I'm just going through a couple of intro slides. But these have been fascinating and really interesting presentations. Sorry. Yeah, cool. Did I mess it up? Oh. Yeah, so hopefully we won't disappoint as the final presentation. And just to get started, I'm going to figure this thing out. Big button. Great. So we don't have any disclosures. We would like to thank Danielle and everyone at the NIJ, though, for having us here and for funding this research. It's in the acknowledgments, but I'd just like to say it on the front end. And we really appreciate it. And, of course, everything presented in this presentation, these opinions are our own. Shout out to the presenter on the necrobiome for pointing out the importance of multidisciplinary research. Now, we realized pretty early on that to successfully complete this project, we were going to need a collaborative, multidisciplinary team. And we partnered with a diverse team from across the United States. And this is because it allowed us to leverage the most innovative and cutting-edge tools to investigate cortical bone changes that affect microscopic fracture morphology within the early postmortem interval, and I say within the first month or two of postmortem interval. So that you can see our team here. We have in Mayo Clinic forensic anthropologists as well as a nuclear physicist in our Department of Radiology who has an expertise in estimating bone mineral density from CT scans. And from ASU, we worked with biomechanical engineers who have expertise in material behavior and structural testing of various materials. And they did a lot of really fascinating analyses from our fracture experiments using high-speed photography and digital image correlation. And then finally, in Mayo Clinic Rochester, we paired up with the bone histomorphometry and core laboratory technicians because they had the equipment necessary to do the histological analyses of bone, and they also had software that could use the digitized slides to analyze basically the mineral and non-mineral content of bone. All right, so usually forensic anthropologists are going to be looking at macroscopic features of fractures to make an assessment of whether or not it's perimortem or postmortem. So for example, fracture patterns. And it's been published ad nauseum in the literature, so I don't need to say a lot about it, but it doesn't work well for us all the time. And the issue is primarily because there's a prolonged perimortem interval in bone. So I'm just saying that bone retains its elasticity, which means it maintains biomechanical integrity longer than other tissues in the body. And this means that perimortem and postmortem fractures often are misclassified. And so taphonomic events can mimic trauma, and then basically macroscopic examinations may yield false positives for perimortem trauma. So given these limitations of macroscopic features for elucidating fracture timing, as in perimortem versus postmortem events, we turn to scanning electron microscopy to examine microscopic features of fracture surfaces of experimentally induced perimortem and postmortem fractures. We documented variables that affect tissue integrity to relate the fracture appearance to biomechanical properties of bone. And these kind of can fall into a couple of different categories. You have variables that are basically innate to every single one of us that are going to vary widely, and I'm talking about bone mineral density and collagen content and cortical bone thickness. And then we have other variables that are definitely influenced by the postmortem interval, like water loss. So we collected all of these variables, and we really wanted to tease out which ones we were kind of driving the features that we were seeing in fracture surfaces throughout the postmortem interval. And our hypothesis was that, obviously, microscopic fracture characteristics may differ significantly as bone elasticity decreases throughout the postmortem interval, which would allow us to discern peri and postmortem events more reliably than macroscopic methods. I'm going to go grab my glasses.
So all of that to summarize, we really realized our experimental design needed to control for as many of those outside variables as possible. So we controlled for temperature, time, and the energy required to initiate fracture. Then we had those, intraspecific variables, things that are varied between each individual and cannot be controlled. So our bone mineral density, cortical thickness, and collagen content was measured and factored into all of our analyses. So this project included 37 unembalmed, defleshed human femoral shafts from males and females, aged 33 to 81, and the periosteum was left intact for all of the bone sections. They were stored at negative 20 degrees Celsius and thawed at ambient temperature or 20 degrees Celsius for 2 days. 8 femora were used as the perimortem sample, and these were not heated at all. Temperature during transport, thawing, and fracture preparation
were used to calculate our observed accumulated degree hours. So, 26 femora were then placed in a Heratherm gravity convection oven to simulate 6 postmortem intervals from 1500 to 40,000 ADH, or 1 to 60 days. A data logger was used to monitor all of our external variables like temperature and humidity, and we weighed the bones before and after heating to estimate water loss. We fractured these femoral shafts at Arizona State University's Computational and Experimental Mechanics facility using a Columbus McKinnon drop test frame. A 3-point bending setup was used with pin-to-pin displacement. So there are a few different options with this. Pin-to-pin is really great because it provides a decent ex vivo estimate of that fixation that you would see for the femur between the acetabular and knee joints in vivo. And then all fracture experiments were conducted with high-speed photography. Four test samples were used to determine the optimum span and drop height with a 15-kilogram blunt impactor. The goal was then to reduce or induce failure via spiral, butterfly, or angled fractures and avoid crushing or extensive comminution. The same parameters were used to initiate each impact and control fracture energy across the experiment. However, we have intersample variability, so we really needed to have a drop height range rather than a single drop height. This was 150 to 275 millimeters. All tests started at 150 millimeters and increased by 50 millimeters until fracture was successful. We then prepared the samples for digital image correlation. This allowed us to document fracture magnitude and directionality for comparison with our macroscopic fracture features and to visualize and quantify those differences at varying postmortem intervals. This video shows the results of DIC in an unheated bone sample. The speckled pattern, as you can see, is spray painted, which allows us to track the displacement of those speckles during the fracture. We then can map cool colors for compression failure and warm colors for tension failure. After fracture, collagen samples from each femur were processed at the Mayo Biomaterials and Histomorphology Core Lab using a Masson Trichrome Goldner stain, which stains for type 1 collagen. Our BioQuant software that we used was really effective in calculating the ratio of mineralized or blue to non-mineralized or red collagenous material in each sample. So as an example, the sample on the left is more highly mineralized than the one on the right, indicated by the greater number of blue pixels. We also used computed tomography or CT scanning to assess the role of relative bone mineral density and cortical bone thickness in fracture mechanics and surface appearance. We mounted the samples in foam and scanned them in a Siemens Somatotum flash CT scanner. Then we calculated bone mineral density from computed tomography scans of the bones using average Hounsfield unit values calculated from ROIs, placed at 90-degree intervals on a cross-section of the shaft. We also collected cortical bone thickness at 90-degree angles around the shaft as well. We also examined fractography to look at that macroscopic fracture characteristic. Fracture surfaces were prepared for fractographic scoring with dual contrast fingerprinting powder and examined under high-powered oblique lighting as recommended by Christensen et al. We created a scoring guide using images and descriptions in Christensen et al. And Love et al. And our scoring sheets contained a femoral shaft cross-section with a mapping template for mapping features, designating areas of interest, like bone hackle, wake features, cantilever curl, and bone mirror. Then we took a look at microscopic fracture surface features with scanning electron microscopy, or SEM. It's really effective for documenting post-mortem microstructural changes, and it has increased resolution compared to light microscopy. We identified the primary tension zone with DIC imaging and produced the looked at the high-speed photography to narrow down our sampling region. We then processed the primary tension zone using critical point dryer and gold palladium sputtering. These images were then captured at a 25 to 30 magnification using a grid system to image the whole sample. Then we identified 3 microscopic fracture characteristics through previous research that the tension zone of the fracture is the best place to observe all 3 of these. So we compared all of these images that we collected and looked for the most osteon-dense image. That was the one we then used for the study. The first feature observed was osteon pullout. It's been found that when substantial force is applied, lamellar structure of osteons deforms to release the stress, resulting in a pulled-out appearance when viewed with SEM. This gives the osteon a telescoped appearance, as seen here. The next feature observed was delamination. Collagen fiber bundles in elastic bone undergo considerable deformation before breaking. This is consistent with the behavior of a ductile material. However, as the bone begins to lose elasticity, collagen fibers begin to have a low deformation threshold, and we see the matrix breaking, micro cracking, and multiple failures in that brittle material, otherwise known as delamination. In higher PMI samples, the brittle interlamellar fibers break, delamination is deep, and the tips of the fibers are sharp, which results in a ragged appearance as seen here. When a critical degree of deformation is reached, we also see the dry matrix actually breaking along cement lines. So we see micro cracks and small fractures along those cement lines. This again is associated with an increased degree of brittleness and a lack of elasticity in the bone. We created 2 millimeter square ROIs in each tension zone image that was selected and used cell counting in Fiji to quantify the features and their relative percentage. Osteon pullout and delamination were quantified by how many osteons exhibited the feature versus total osteons. For delamination, the osteon was counted if greater than 50% of the osteonal lamellae exhibited delamination. Microfractures were simply quantified. We also wanted to note that it's very important to generate the correct lighting angle under SEM testing conditions because these allow us to reduce image flattening and get the most accurate view to assess osteon pullout. We screened our data for a degree of curvilinearity using lowest regression. Lowest regression indicated that linear models sufficed for our data. Multiple linear regression was used to examine the relationships between our quote-unquote tissue integrity variables, so ADH, water loss, collagen percentage, bone mineral density, and cortical bone thickness, and our microscopic fracture features, so osteon pullout, delamination, and microfractures. We used Pearson's correlation to test the relationship between ADH, water loss, collagen percentage, donor age, and fracture energy variables, and microscopic fracture characteristics. All of our statistical analyses were performed using R and RStudio, and our plots were done using the shiny new ggplot GUI.
All right, so the first slide on results presents just a brief summary of the macroscopic fracture assessment. We presented this last year and it has been published in Journal of Forensic Sciences, so I won't go into it in detail here. We're focusing on the microscopic features. But basically, the fractographic features were similar in our experimentally induced fractures throughout the postmortem interval that we looked at. So this bodes really well for fractography's utility, but it underscores the capacity or limited capacity of macroscopic fracture morphology for distinguishing perimortem and postmortem events. Now this table Just not to bombard you with tables, I'm going to spend a little bit of time on this one. So if we look at how well the variables that we selected, the features that we selected, indicate the timing of a fracture in the postmortem interval, using the whole sample, our adjusted R-square is 0.65. And so with that, I would say that we had most of our sample between perimortem and 16,000 ADH, and that's up to about 20 days, okay? Then we had 3 extra samples thrown our way, and so we decided to see what would happen if we basically doubled the postmortem interval. So we had 3 at 40,000 ADH, and that's a couple of months. And when those 3 are removed from the model, this R-square jumps up to 0.91. Okay, so there's a gap in what's going on, after the postmortem from basically 1 to 2 months, okay? And that's where we'd like to focus our next efforts and see what's going on there. Early in the postmortem interval, these features account really well for fracture timing. And actually interesting is that just osteon pullout gives an R-square of 0.87. So it's an excellent indicator and probably our strongest one. Let's see. Sorry. Great. Okay, so if we look at all of the variables that we mentioned that affect the biomechanical integrity of bone, we throw them into a regression model and do stepwise selection and see which ones are accounting for the variation in each one of those features. The one that comes out in every single model is water loss. Okay, so early in the postmortem interval, I know if you read the literature, everything talks about collagen. It's not collagen, it's water, okay? Bone has a ton of water in it. We'll talk about it in a minute, okay? Collagen certainly is going to come into play later. Early on, we're looking at water. So just taking a look at these as the perimortem interval increases, as water loss increases, we're going to see a decrease in that osteon pullout. So osteon pullout is a signature of elastic bone, okay, as we would expect. Those 3 little dots that are always up there, guess which ones? 40,000 ADH, okay? We didn't take any data out of our models. I mean, it's nice, it makes pretty models when you screen for outliers, but this is biology, y'all. I mean, we can't do that, okay? There's no perfect human being to undergo fracture experiments. So we kept all this data in because I think that's important. And delamination, so as postmortem interval increases, water loss increases, we see an increase in delamination. So that's a signature of bone that's becoming more brittle. And this microfractures, as a variable, interestingly, I think it's our weakest predictor, but there still is a correlation, and you're going to just tend to see more microfractures as the postmortem interval is increasing. This is a correlation matrix. Red is a negative correlation, blue is a positive correlation. And the strength of the correlation is shown by the size and deepness of color of each of the circles. So if you look at the white asterisks, what it highlights here is that the variables that we selected, so osteon pullout, delamination, microfractures, they correlate very strongly with postmortem interval as calculated by accumulated degree hours. And then water loss, the same. Okay, because like I said, what we're really, but look at the other variables. Look at collagen and look at, that they're just not showing up in our early models. Sorry, I know that.
So something else we wanted to start this discussion with is, as we found out through our fractographic analysis, we see a lot of consistency, especially in the early postmortem interval, in those macroscopic characteristics. So, anything from 3,000 to 16,000 ADH have nearly identical macroscopic fracture characteristics. And then our microscopic features change significantly. So we really see those mapping onto ADH, and we see a very clear delineation. When we look at the different microscopic fracture characteristics of bone, and we look at those mechanical characteristics, we really see that elasticity affecting bone very highly in the early postmortem interval, especially seems to be associated with things like water loss. So here's our early PMI versus our late PMI. We see a lot of difference here. Same thing with delamination. We see a greater degree of elasticity in the fibers, collagen fibers, on the right, and a much reduced degree of elasticity on my left, not yours. Once we get into that prolonged postmortem interval, we have some other examples of what's happening with collagen. So,
yeah, so collagen. So this is, I think, what we did was we looked at collagen. Collagen is highly variable between individuals. It's like bone mineral density. I mean, it's highly variable. So we wanted to quantify it, and then we were like, well, let's see what's the change in collagen throughout our postmortem interval. And what this shows in these samples is that, again, up to 16,000 ADH. So the first 20 days or so. Is collagen decreasing a little bit, but not significantly? But take a look at 40,000. So as we move through the postmortem interval and the water within the bone is liberated, collagen then is, I think, the next thing that's affected. So we're not going to see collagen playing a significant role, I think, in contributing to these fracture surface characteristics, other than the fact that it just maintains elasticity, right? So water is going to be our big driver if we're seeing any elastic changes in the early postmortem interval. So excuse me while I nerd out a little bit on the extracellular matrix, but I think you have to understand this to understand what's going on. So if you look at this picture, the top part is outside of the cell, extracellular matrix. We have these big pink cross-linked collagen fibers, and then we have these little things that look like trees, and those are glycosaminoglycans. So we've got free water in the bone, in the Haversian canals, and it's not bound to anything. But then we have some loosely bound water that's bound to these glycosaminoglycans or GAGs. And then we've got so about maybe 30 to 40% is free water, maybe 40 to 50 or 60% is loosely bound water, and then the very little bit left is going to be tightly bound water into collagen molecules. So you can kind of see how water is liberated, free water first, then the loosely bound water to the GAGs, and then finally, it's going to be that tightly bound water in the collagen.
So because of this, we were able to accept our hypothesis. We have these microscopic fracture surfaces that are really diagnostic so far. And it's very interesting because they're very connected to the hydration of bone and some of those very interesting mechanical qualities of bone. And then, finally, we're able to have a lot more sensitivity in this type of analysis. Sorry, I accidentally clicked too fast. Our power analysis suggests we need a larger sample size, of course, as always. We really need to be able to look at a greater span of ADH. We need to look at a greater range of individuals, of course. And we also need to calibrate the data in some additional ways. So we need more bone density studies, pre and post heat, I think, collagen analyses, pre and post heat, and additional SEM evaluation and validation. Then inter-observer error and machine learning to streamline this assessment process. And then we're also investigating the use of micro-CT, which can be leveraged for this type of analysis and maybe a little bit more accessible depending on which crime lab you're at. So we were able to accept our hypothesis, and we welcome any questions. Thank you.
A couple of related questions. You worked with defleshed bone in this study, but how would you predict fracture mechanics might change when muscle and fat are present? And does the defleshing process impact the structural integrity of the bone in any way?
Right, so most of the studies in this, in order to do high-speed photography and see a sample, we have to work with defleshed bone unless we're going to be using some type of X-ray photography or something like that, which would be the only way I could think of to get at that, yeah. So it would be a totally different setup, and it would be harder.
Last question, since water loss was a major predictor of fracture features, would you predict different outcomes in a warmer, more humid environment?
I think you would just predict that the water loss would occur at a different rate. And by the way, our environment was warm. So the oven was actually warm. And then as water was liberated, it created quite a humid environment. We did use a data logger to record that data. So I think for me, the bigger question is going to be what's going to happen in a cold, dry environment. But it's just basically going to, it'll affect the rate at which the water is liberated. And I'd just like to make one comment about animal versus human bone, because I think we have received that question. If we're looking at osteons and osteon pullout, we have to use human bone. So I'd just like to put that to bed, because that coming up is one of the most important variables for us. I think was, we've received some comments like, wouldn't it be more, cheaper to use animal bone and to be able to get a bigger sample? And we would love that. But I think we can't really look at I don't know how to I just don't know how to translate osteon population densities when we're looking at. So I would love to. But with that, I would also invite anyone interested in this work to please contact us. We would love to collaborate. If there's anything you have to offer to us that we have not thought of, we would love to collaborate with anyone here or anyone you know of. So please reach out to us. Thank you.
Pause for our presenters for this last session before lunch. Thank you all, and we will reconvene at 1:25 with Seas, Drugs, and Toxicology with Francis.